Last time we finished the general sense. Today we're in the spatial senses. Spatial senses include the smell, the taste, vision, and auditory. First, smell. Smells is detected by the olfactory system. Detect the airborne molecule. This molecule need to go into the nasal cavity, and on the upper part of the nasal cavity being picked up by the receptor. These slides tell you the epithelium cells of the olfactory system. These are the receptor called olfactory epithelium. And the chemical molecules will come to bind with the receptor, and it will transfer those physical stimulus into the neurotransmitter release and action potential. Olfactory system connect with your limbic system in your brain. The function of the limbic system is memory and emotion, like amygdala is associated with the emotion. Hippocampus, that's the memory. Because of this connection, olfactory is a big trigger of memory, all memory, emotion-related memory. And that's when you smell a food and you turn to tell your friend, my grandma used to make this kind of food. And those old memory, 10 years old memory come back. And because of this anatomical connection. The other kind of chemical sense is called the gustatory. Gustatory system detect a chemical molecule dissolved in water and create flavor. There are 4 traditional flavors: sour, bitter, salty, and sweet. And there is one more new flavor, and it's called umami. So totally 5 different kind of flavor. Umami is a savory flavor from amino acid glutamate. You can find it in the broth, meat broth, and also fresh seafood if you like sashimi, and that's because of umami. Now let's go to the visual system. The eyes include 3 layers. Fibrous tunic, that's the strong tough layer, protect the outside. Vascular tunic, that's the middle layer with blood vessel. And the inner layer is called the neural tunic, and that's your retina. These slides show you those 3 layers, the outer layers. Fibrous tunic, including the cornea, that's the transparent part, and sclera. Sclera is the tough opaque layer, protect the eyeball structure. And the second layer called vascular tunica, you have the iris, you have give you the eye color, also provide the opening called the pupil. And the ciliary body, that's the muscle to pull the lens and change the curvature of the lens, so you are able to see the object close or far away from you. The third layer is called the neural tunica, and the retina, that's the third layer. There's a piece of paper in the end of the eyeball, and that's the most important structure in the eye. They transfer the light into the neuronal signal. These slides show you the outside structure of the eyeball, and the outside structure of the eyeball including the lacrimal apparatus, that's the tear gland, and it produces tear. When they produce tear, it goes from the lacrimal gland is on the lateral side. You watch the eyeball and go to the nasal part. So when you cry, you feel like the tear go into your nose, and that's because of this movement. These slides show you your eyeball structure, and the lights move in through the cornea and reach the pupil. Pupil is opening created by iris. There's no anatomical structure of pupil, it's just the opening. And this, when you dissect the eyeball, I call this mushroom, and this is the ciliary body. And the lights will move in, hit the retina. On the retina, the center part called fovea. Fovea is the part give you the sharpest vision. You have clear vision. That's because of fovea. And eventually, the signal will be transferred into neuronal signal, goes to the optic nerve, cranial nerve number 2, and you will go back to your brain. Because you only have a very small sharp area called fovea, if you want to see clearly, you have to move your eye to the target. So outside of your eye, you have 6 muscles. 4 rectus muscle, 2 oblique muscles. So this muscle will pull the eye and move the eyes to the correct position so you're able to see clearly and make the object fall on the fovea. That's another view to show you those 6 muscles: superior, inferior rectus, medial, lateral rectus. So they pull the eye up, down, medial, lateral, and 2 oblique muscle. When they pull the eye can rotate. And these 6 muscles need to be coordinated well, and also both eyes, 6 muscles need to be coordinated well. And that's why in the cranial nerves, cranial nerve number 3, 4, and 6 were dedicated for the eye movement. When they were not coordinated well, and you will have the eye tilt a little bit compared with the other one. Usually happens in number 6, abducens. The inner layer of the eye is called the neural tunic, also called retina. That's the most important structure of the eye. It transfers the light into the neuronal signal. So that's in the inner layer. And when the lights move in from the outside, it will go through the cornea. That's the transparent part. It will go through the lens, and now you'll reach this part called the fovea. That's the one give you the sharpest vision. This is a picture taken from the front view of the eye. So you take a picture, go through the pupil, that's how you look like. And this area, that's the fovea, give you the sharpest vision. That's the area called the optic disc. It also kind of called the blind spot, because in this area you won't be able to put any photoreceptor there. That's the input and output of blood vessel and nerve. So you could not put the photoreceptor there. Because of that, and no photoreceptor, no vision. That's a blind spot. So this is where the optic disc, the blind spot, is located. So that's where the blood vessel move in and out, and also the nerves, cranial nerve number 2, move out. And the retina, let's look at the structure of the retina. You have the photoreceptor, and you also have the bipolar cells and ganglia cells. We won't talk about the amacrine horizontal cells too much. Together, the output is called the optic nerve, cranial nerve number 2. That's the structure of the retina. So you have the photoreceptor layer, it connects to the bipolar connect to the ganglia cells, generate the action potential and send it to the brain. You have 2 kinds of photoreceptor, the rods and the cones. The rods are the one very sensitive to light, but you only have one kind of rod. Because of that, no matter what kind of wavelengths of light hit the rods, they give you exactly the same response. So it can only create black and white, no color vision. And we use them in the night. The cone systems need more light to activate them. We have 3 kinds of cones, and different wavelengths will activate different cones. Because of that, they are for color vision. And in the fovea area, the area with the sharp vision, they're all cones. We have 3 kinds of cones, and the way it works is like the computer monitor, each pixel, they're created by the red pixel, the green pixel, and the blue pixel. They just change the relative intensity of these 3 different kind of color and give you the whole spectrum of the color. So human, we have 3 kind of cones. Compared with other species, we are good but not the best. We are better than dog. Dog only have 2 kind of cones. And compared with bird has 4 kind of cones. So they actually have a better visual system than us. And some species like this, the mantle shrimp, actually have 12 different kind of cones. And humans have the three kind of cones, and this mantle shrimp have 12. So it covers the whole UV till the infrared range. And let's take a short break.